Okay, I can see myself.
So just starting.
Yeah, yeah, sure. Please do.
Shall I start, sir?
Yes, 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 please.
Okay. Good afternoon. We are presenting on the topic of heterochronic parabiosis. So, at first, we need to know the term parabiosis. It refers to the condition in which two living organisms are joined surgically, sharing a common circulatory system termed as anastomosis. It is a complete form of transbiosis. Unlike transbiosis, there are no formal donor and host in parabiosis, as each organism viewed as equal partner. In simple terms, it is analogous to anomalic condition in different living organisms called conjoined individuals, known as Siamese twins in humans. There are two types of parabiosis. One is heterochronic parabiosis, where it involves organism of different age. Another one is isochronic parabiosis, where it involves organism of same age. Now, please have a look on this pictorial depiction.
The significant milestones are shown in this. However, the most important milestone was the first experiment conducted on heterochronic parabiosis on mice in 1904 by Nobel laureate Alexis Carrel.
From several studies, scientists discovered various aspects of aging. Then the most potent question was whether the decline in regenerative potential is due to irreversible age-related changes in stem cells or by alteration in microenvironment. The solution was provided by studies of heterochronic parabiosis in the following year. To address this question, the experiment was began by using three sets of parabiotic pairing, where one was heterochronic parabiome involving young mice and old mice, and other two sets are of isochronic control, where one is a pair of old mice and other is a pair of young mice. Now, the procedure begins by giving anesthesia to individual mice and prepared them for surgery, where a skin incision was made along the opposite flanks of each mouse and stitched them, waiting for the formation of shared circulatory system. Now, the post-surgical care is the most vital phase in this experiment, as because 50% of the paraband die before the formation of complete anastomosis in 2 to 3 weeks due to the parabiotic intoxication or parabiotic disease. But due to the improved anesthesia and post-operative monitoring, 90% of the pairs recover from the procedure and are examined after reversing the parabiotic pairing.
So as we learned, it takes almost 3 weeks for complete anastomosis. So after a time period of 1 month of parabiosis, it was observed that the older heterochronic parabiome had a better rejuvenated tissue regeneration system, a better neurogenesis, reconnection of cerebral hippocampus, increased neurons and stimulation of synapses, reactivated cell and growth factors like GDF11, NANF, faster muscle repairs, enhanced skeletal muscle functions and cardiac function, improved sensory perception, that is an overall rejuvenation of the stem cell. From the comparative studies with the isochronic controls, it was observed that the heterochronic fold has got more mitochondrial area in the skeletal muscles than an old isochronic, as well as better respiratory functions, that is better oxygen flux by the skeletal muscles. While the younger heterochronic paradigm had a decline in neurogenesis, muscle function, tissue regeneration, and stem cell regeneration potential. Also, it had increased amount of CCL11 that decreases neurogenesis and cognitive performance. Hence, it was concluded that young blood provided a younger environment to relatively old cells and tissue, causing enhanced phenotypic and biological performance in the old paradigm, while the old blood caused decline in biological performance of the young paradigm. Thus, it was proved that aging depends on the microenvironment of the stem cells. On the other hand, dead cells and old blood is noxious and causes difficulty in dissection. Hence, causing the acid and why many stem cells are become methods resulted in an extended lifespan of the subject. Hence, the uh, that and vice versa.
Arnav, you are sparsely audible. Can Vidipta take you from here? Vidipta, can you take it from here? I think you are audible
Yes,
well.
sir.
Okay, I
Yes,
think it's
sir.
over
Sir, can
already.
I?
You can just explain the, you are already, you people are on time. Just explain the result slide just once in a very brief way. Just Vidipta.
Okay, okay. Okay, okay. So basically, that we are getting, that we are observing that the older heterochronic paraband had a better rejuvenated tissue regenerative and neurogenesis reconnection of cerebral hippocampus as compared to the isochronic control. And from the another observation, the heterochronic old has got more mitochondrial area in skeletal muscle than the isochronic old, and as well as a better respirometry of skeletal muscles. So,
Yes.
and it was proved that aging depends on the microenvironment of the stem cells. On the other hand, dead cells and old blood is noxious and causes difficulties in differential of cells, hence causing age-related issues and rapid degrowth. This also explains why stem cell transfers is unpromising. Thus, later a clinical procedure was developed, that is HPB, that is heterochronic parabiosis protocol, which combines the blood purification, young plasma exchange, and stem cells transplantation. So this results in an extended lifespan, hence it promotes reverse aging.
Okay. Okay. I think
But I
anybody from the
guess
panel would like.
she missed a part. She missed a part from the negative impacts on the younger paradigm. Can you, Vidita, can you please explain that part?
Arnav, you are sparsely audible actually. Arnav, you may switch off your mic. I would like to request any panelist if they would like to ask something. We have uh, Shashwato, maybe Anonna, anybody? Anonna is here from Anonna and Agrudipa from Germany right now. Any questions or suggestions? Orko?
I only have a few suggestions for you regarding the presentation. Content-wise, it was good. It's good that you presented more data-related stuff rather than just texts. But I would suggest that maybe spread out the data over a few more slides, or don't show this much data because all of your slides are very information packed, and that tends to lose the crowd's attention. So keep it
Okay.
informational, keep it data related, but don't overcrowd slides. Focus more on explaining or interpreting those data when you are presenting. And also, when you're presenting, don't expect people to understand pictorial depictions on their own. Instead, what you can do is the pictures that you have given on the slides, you can use them as a reference while explaining that particular point. Other than that, it was very nice. Thank you.
Thank you.
Orko? Okay.
Yeah, I had same suggestion. Like, if you're presenting data from a paper, like, try to like show one figure or one experiment per slide. So That's and also used
Okay.
for like figure legends and like a few points under the figure so that people can understand the figure. And yeah, that's all. Like, yeah, same as Ananya mentioned, it's like a little overcrowded the slides. Yeah, but yeah, good job.
Thank you.
And one very small suggestion, please, whenever you are presenting scientific data and everything, please put page like slide numbers and everything. These are the small mistakes that will be asked for later. Yes.
Also, another
Uh,
suggestion from my side, which is a very small uh, suggestion, that use pointers. And when you are presenting, use the laser pointer to show the audience exactly what you are speaking about. So there's a lot of data, as Anya already pointed out, that there's a lot of information. So even if that's the case, if you use a pointer, then the audience can focus, okay, this is the one that they're talking about. So it's just a small suggestion. Overall, nice presentation, and yeah, try uh, what to say, condensing the data to something more relatable to the audience, because as panelists we don't have any prior knowledge to these things, so you have to be as simple as possible. So yeah, avoid jargons, try making us understand things with much simpler terms. So yeah, otherwise it's a good presentation. Yeah, thank you.
okay, sir. Thank you.
From my side, it has been really, really nice. I really liked your, you have utilized your slide space very nicely. The comments that has been given by everybody, that is true. Just one more addition. If you while you're explaining so many slides which has so much things, then it is better if you give a one-way takeaway, a one-line takeaway. If you have in each slide, then it is easier for us to know that what we are expecting from this slide. So if that is all, but given your experience, it has been fantastic, really nice. I mean, thank you very much for presenting. You may stop, and we have Vedatri and Devnarayan. I guess they also have a commitment today. I think they will present.
Sir, sir, I am leaving,
Yes.
sir. Actually, my
Yeah. Yeah.
syllabus
Sure.
got
Sure.
stuck right now.
Please. Yeah.
Thank you.
Fine. Vedatri and are they here?
Yes, sir.
Yes,
Yes, sir.
sir.
I'm audible.
Yeah. Please.
Is the screen visible?
Yeah. Yeah. It seems to be a very interesting one presentation.
Shall I start, sir?
Yes. Yes. Yes.
Good afternoon, everybody. I, Vedatri Patanayak, along with Devnarayan Das, we would like to present the research study about the impact of depression and anxiety treatment on biological aging and metabolic stress, study of protocol of the mood treatment with antidepressants or running study. This research was done by Lever Van Mulligan et al. In the year 2019. Could you please show the next slide? Thank you. Depression and anxiety disorders are common among public health. It is characterized by persistent sadness, hopelessness, and a sense of unease or fear. It can impact daily life, relationships, overall well-being. Treatment options include therapy, medication, and lifestyle changes. Um, could you show the next slide, please? I'm so sorry. Is it? Um, Anxiety and depression have been found to impact a multitude of aging-related somatic conditions. Shorter telomeres have been found in depressed and anxious patients as compared to healthy controls.
Telomere length is associated with mortality. Telomeres are tandem repeated DNA sequences that protect the chromosome ends. Telomerase activity is the deciding factor for telomere length. Higher telomerase activity prevents rapid aging and cell death, whereas lower telomerase activity leads to aging-related disease factors. In this study, we have observed abnormal telomerase activity. Also, studies have shown depressive and anxious people have increased risk of cardiovascular diseases, diabetes, stroke, also advanced physical and cognitive decline. This in turn affects aging. Some other studies show depression and anxiety to be linked with systemic inflammation, oxidative stress, hyperactivity of hypothalamus pituitary adrenaline axis, and metabolic syndrome dysregulations. For the purpose of this study, two ways of treatment have been chosen. Could you please show the next slide? Am I audible?
Yes, you are audible, but I think there's.
Have we lost Devnarayan in the process? I don't know.
I think he's having network issues.
While you are presenting, since online presentations are now a thing, please use Microsoft 365 or Google Slides. Yeah, we are back with the three. You can continue.
Thank you, sir. For the purpose of the study, two ways of treatment have been chosen, one by antidepressants and another by running therapy. Commonly prescribed antidepressants are SSRI, which is selective serotonin reuptake inhibitors. These work by increasing the levels of serotonin in the brain. Patients participating in this study were prescribed 10 milligram of escitalopram per day. However, some patients were not responding to this SSRI. So another SSRI was prescribed for sertraline, 50 milligram per day. Both of these SSRI have higher efficacy and favorable side effects. Similarly, running therapy was prescribed to another group of patients. 3 sets of 15-minute 45-minute outdoor running session was prescribed per week. They started with a 10-minute warm-up session, then 30-minute jogging at an intensity that maintains the heart rate, and the running session was ended by a 5-minute cooling down exercise. Total 160 patients were chosen of an age range of 18 to 70. It was a 16-week intervention study. Patients were grouped by an SPSS random generator into two groups. One group underwent running therapy and another group underwent antidepressant therapy. 50 patients were subjected to MRI. 25 patients from each group were were subjected to MRI. No disease, no treatment control group were examined to compare health, physiological, and neurobiological indicators with healthy control. Thank you. Now, my partner would explain the rest of the presentation.
Thank you, Vidatri. Good afternoon, everybody. I'm Dev Narayan Das, and I would be explaining the rest of the paper. Getting back on the topic, we have eligibility criteria. Patients were screened before they were allowed to participate in the study. The inclusion criteria were having depressive and anxiety disorder, and their age should have been between 18 to 70 years of age. The exclusion criteria for the same were antidepressant usage should not have been there within the last 2 weeks. Current usage of psychotropic medication, except for the usage of benzodiazepines with stable usage, regular exercising more than once a week, clinically diagnosed depressive or anxiety disorder, evidence of acute suicidal risk,
medical contraindications to running therapy of antidepressants, or being pregnant. The patients for no disease and no control group were chosen carefully. The inclusion criteria for the same were having negative lifetime history of psychiatric disorders, and their ages should have been between 18 to 70 years of age. The exclusion criteria for the same were they should not exercise more than once a week, and the medical contraindications to running therapy and antidepressants should not have been there. Now, to assess the outcomes of the study conducted, various tests were done to check for signs that would help in making a firm opinion on the result. The primary outcome, of course, being the change in biological aging that was measured through telomere length and telomerase activity, which we know have direct relations with aging. On the other hand, secondary outcomes that were assessed during the study were biomarkers of physiological health, which are gathered through fasting blood samples, urine, and 6 saliva samples that were taken at different time intervals. Then we have presence of depressive and anxiety disorder, which were established by using CIDI, that is Composite International Diagnostic Interview, which also using methods and tools such as life chart method, Beck Anxiety Inventory, Fear Questionnaire to assess the severity of their disorder. Lifestyle indicators, which relate to change in healthcare status and utilization, personality, and cognitive vulnerabilities, are defined through various questionnaires such as Squash Questionnaire, Neo FFI Questionnaire, and Leeds R Questionnaire. Neuroimaging assessment. Here, a subsample of patients and healthy controls underwent some tests to assess their verbal episodic memory, which was done by the 15 words test, their task related brain activity, which was done by anatomical T1 weighted and diffusion tensor imaging, and finally, brain network connectivity, which was done by functional MRI images during rest. To conclude the research paper, both treatment methods have shown to be effective in depression and anxiety, but a well-designed comparative study of these treatment strategies and their impact on physiological and neurobiological process is currently lacking. As the results obtained and observed after the study show that both the treatments are equally good and none is better than the other, not only these two, a wide range of treatment programs are available, but it remains largely unknown whether the impact of these programs are comparable. This particular study aims at providing more information about the underlying biological mechanisms of depression and anxiety treatments that would be helpful in increasing the effectiveness of personalized medicine. These were the resources that were used during the process of making this presentation. Thank you. That would be all from our side. We are open for questions.
Yeah, thank you, both of you. What's the conclusion? Can you just tell me? I lost my connection a while ago. So
So basically, the conclusion is like we cannot say for sure that one type of therapy is better than the other, and there are still
Yes.
studies yet to be conducted in this topic.
what about antidepressants?
Uh, the results seen in both the cases have been very similar. There is not much difference in both the results obtained after running therapy and antidepressants. So
You know the sample
basically,
size of this? But this was a clinical study, right?
yes.
What was the sample
Uh,
size?
The sample size was of 160 patients, including a total of 160 people, including 100 patients with anxiety or depressive disorders and 60 healthy controls, which did not have any history of having any psychiatric diseases.
Okay. Anything else from the panelists?
Yeah, I
Hi.
have.
Okay,
Yeah,
Orko, you can go first.
okay, okay. So I have a few questions. Can you go 2 slides back?
Sure.
Yeah. Yeah, the previous slide. So, have they mentioned like what are the biomarkers of physiological health that they have taken into consideration?
So they did, but they are testing for basically cortisol levels, and yeah, they were testing for cortisol levels, metabolic syndrome abnormalities, DNA and oxidative stress, and inflammation markers.
Okay. And you mentioned quite a few times about the altered or telomerase activity in patients with depression or anxiety. So, like, I mean, which cells have they considered? Like, telomerase activity is not present in any somatic cells, right? It's only there in stem cells. So,
I don't
like,
think they
like
specified
during the
which
assay,
cells
like,
they.
which cells have they taken into consideration? Like when they mention about the telomerase activity.
It's not specified. They have just said somatic cells, I guess. Yeah, Vedatri, go ahead.
So they have not specified which cell they have used. It's just mentioned that they have studied the telomerase length and they have found that is shortened. And telomerase activity is also sometimes in some patients it has shown that it is elevated, and some patients have shown decreased levels of activity.
So
But it's
these
not.
are in the like mesenchymal stem cells, right? Or in the gametes, like because like I'm just for your information, like telomerase is only present in stem cells or gametes, or if you have cancer. So it's like somatic cells don't have telomerase. So yeah, I think they have then just taken into consideration the telomerase activity of the stem cells. Yeah, okay, that's all. Thank you.
Thanks, sir.
Thank you.
Agradi.
Hi. I just wanted to ask, was this a review paper or this was a research paper? Eh,
Uh, this was a research paper, if I'm not wrong.
just one suggestion from my side is that if you're presenting research papers, uh, they should have some data in them, maybe graphs, bar graphs, histograms, whatever. You include them in your presentation so that it's clearer to the audience that, okay, this is what they found, because your PPT is nice. It's very well organized, but if you had include those images that they have prepared, maybe some graphs, some other figures, I don't know what they have in their paper, that would have been much clearer. Because whenever you are presenting a paper, even when I do present the paper, usually I include those figures. That's a very important part of presenting a paper. You cannot just skip their images and you give a like a overall gist. You have to present the data, but not like cram your slides with data. Just a smart way of presenting, right?
So, the
Sure.
paper did not have any charts or
Did not have okay,
graphs.
okay,
They
then
had
It's
I mean, they had a chart of what tests they did on the patients, but that was it had mostly no data. It represented in which
okay.
weeks they have taken the sample, but that was it. That was the only chart. It had a lot
Okay.
of questions and things they had taken from the patient.
Mm,
That we have
okay, okay.
mentioned in.
Yeah, but otherwise your slides are very nicely made. I like them. Yeah, and nice presentation, guys. Good job.
Thank you, sir.
Ogradita, I think sorry.
Go ahead, go ahead.
Hmm.
I think the paper they referred to was just a study protocol rather than the study itself. So
Okay.
it mentioned only what they're going to do and the procedure rather than the study and the records and results
Yeah.
and all of that.
Yes,
But
because
yeah,
when
for
Orko asked, yeah, when Orko asked the same thing, I was a bit confused because usually they will have to mention
mention
which
the
cells they
type, yes,
took for
exactly.
the studies. Yeah.
But for you guys. I would suggest it's, and this is also another like presentation suggestion. It's very good, but I'm just suggesting you points that can make it better. Firstly, for the first slide, you explained what depression and anxiety is, and that is okay, but maybe use statistical graph or something that shows some kind of data driven image that shows the level of depression and anxiety that is globally present right now or is affecting the entire world in different proportions, and then use that image to explain what depression and anxiety is rather than just putting the definition and reading it out from the slide. That is one point. Another point is you can structure your slides a bit better and keep them in proportions. Like don't keep slides where there's like one text in the middle and the other text is a little below that when they are like side by side and comparable. And also, can you go one or two slides ahead? I don't remember which slide it was, but I'll tell you. Ahead. Yeah, for this one, like see the antidepressants line and the running therapy line are not in line, firstly. Secondly, you can also use, like when you are putting comparable studies or two different studies, you can also use a table so that it looks better. And there's not a lot of white random space in between. And also that line anxiety and depression can be treated by, that can be in one line instead of just treated by in the second line. These are very minute things that I'm saying. And also for the last presentation as well as this presentation, try to keep borders on every slide and try to keep equal borders on every slide, because then it appeals to the eye of the beholder. That's it from my side. But good presentation. I think you guys understood what the study was about, and that is the most important thing. But for future, if you are referring to a paper that is only giving you the protocol and not data driven results, then I would suggest that see where these papers are taking their references from and then go to those papers to see if there are like additional studies or additional information that you can find there. It will help you to understand the data better as well, and also like make your presentation better, I think.
Thank you. We will keep that in mind. Thank you.
I have a very small question. Like you mentioned, they did neurobiological imaging and everything. So, are there any particular areas of the brain that they map in order to assess the study, or like mentioned whether they will map this region or these components?
No, sir, they did not specify exactly. They have just said gray and white matter structure. That is all they have said.
Okay, okay. Uh, like, are you sure this paper did not have any supplementary data either?
So now that I notice, it is a study protocol. It is written right
Okay,
above topic.
okay, okay, okay. Okay, it's fine. Otherwise,
Okay, I
it's a good
think it's fine.
presentation.
Uh, no suggestions from my end. Actually, people from various fields, they're giving their expertise. So I really do not have much to say on this. Just it was a good presentation. I'm really proud of you people. Thank you. I think uh, now we can take up the panelists would present in between. So it will be starting with me. I'll present another one of my own papers, and then we'll go with maybe Agrideep and other people. And you people can just watch, and then we will start again with your presentations. So this had to happen at first, but anyway, this didn't happen as we planned. So is the slide visible?
Yeah, this.
Yes, it's visible.
Okay, so this is just give me a moment. I'll have to find like pen. Yeah, so it's a study of Just a minute. Yeah, selective depletion of ZAP binding CpG motifs in hepatitis C virus evolution. So, regarding hepatitis C virus, it's a positively sense RNA virus belonging to the family of Flaviviridae. It causes chronic liver diseases and cirrhosis. Also, it has some links with, in developing liver cancer. So what studies have shown already is that there is a depletion in CpG content. CpG is nothing but C's and G's. So CpG, they reduced considerably in viruses that affect humans. So ZAP, which is zinc finger antiviral protein, it binds to these CpG motifs and it restricts its replication. That's the idea. And one paper, they also give what is the optimal binding site for these ZAP proteins. Now, this is how the virus looks like, and it essentially in its protein coding region, it has three structural proteins: the capsid and enveloped glycoproteins. And we will see about these proteins later in this study. So regarding ZAP mediated viral restriction, how it works: ZAP is a protein which binds to the DNA, which has DNA and specifically RNAs which have more CpG content. So if a virus has higher CpG content, it will be restricted, it will be degraded, it will not replicate. And if a virus has a lower CpG content, then it will be able to replicate, and that will essentially, drive the evolution pattern. So some of the results of this study. Before we get into that, there were certain things which we did. So we took 2,616 sequences of hepatitis C virus full length sequences, and we analyzed the number of CpGs and the ZAP, and all this was done using BioPython. And one another thing was that all the sequences before 2002 were marked as historical sequences, and after 2010 they were termed as contemporary sequences. So that will be helpful to understand the graph. So as you can see, the number of CpGs, they have reduced in the contemporary sequences compared to the historical sequences. And if you see the timeline between 2001 to 18, there is an abrupt increase right here near 2014, 15, 16. It's very interesting that same pattern was also observed for the number of ZAP binding motifs, which are usually observed. There also there is a sharp increase at during the same time. And it was merely it couldn't be a coincidence, but during this time, direct action antivirals were also introduced for hepatitis C virus, which reduced the virus load in the blood to a great extent. So at the same time, now this CpG loss could be due to loss of GC content also. So it could be because GC Cs and Gs are reducing. That's why CpGs are also reducing. So to know understand whether CpGs are reducing due to the loss of GC content alone, we did something called CpGs O by E. That is the observed CpG numbers, the change in observed CpG numbers compared with their expected numbers, which is the GC content. If we divide this, then also we see a very faint, increase near a similar pattern is observed, and statistically a reduction in CpG O by E is also observed. So when the CpG numbers and the ZAP binding motifs, they were plotted against each other, there was a direct correlation, a positive correlation. So which states that if there are more CpG numbers in a viral genome, it has the higher propensity to be degraded by ZAP mediated restriction. So uh, so within ZAP binding motifs, so these ZAP binding motifs are the specific sequences which contain CpG. So we had to see whether CpG is lost only within the ZAP binding motifs, or it is lost up outside also. I mean, the loss is random, not because of ZAP binding motif or outside. So we saw that the loss of CpGs within the ZAP binding motif is pretty significant. And it's remarkable in a sense that we can say that ZAP mediated restriction was the driving force for loss of CPGs within the viral genome. In this case, you can see the presence of the conservation of different ZAP binding motifs. These are the ZAP binding motifs, optimal ZAP binding motifs, which can have uh, like CN4, then G, and then another nucleotide, and then there are CGs. So CPGs are present. From 4 to 8 nucleotides, these ZAP binding motifs could be there. And the interesting part of this analysis was that most of the ZAP binding motifs, they were concentrated at the core region of the viral genome, while other places it was sparsely populated. So it is interesting to see that the core region of the virus, it maintains its sequence, and it is not that mutable. So that is one finding, very interesting finding from this paper. And then we found that what is the CPG loss within the core protein region. There also we see that within core protein region, there is a CPG loss. But even with that, the CPG numbers, if you see within the core protein region compared with the rest, then the CPG numbers are pretty much high in the core protein region. So that's what we observed. So um, this actually study, it's a very short study which tells us about the pattern of viral evolution with respect to CPG depletion. And again, if we introduce some other kind of stress like direct action antivirals or maybe vaccines, this declining CPG trend is sometimes reversed. And one important message from this entire study was that ZAP-mediated selection pressure, it drives the CPG evolution, depletion in HCV genome. However, the core protein of HCV, it is not restricted by ZAP-mediated depletion. So that's the study all about. Is there any question from the audience? No
Sir,
question?
sir, can I ask one question?
Yeah, sure. Mayuk.
Sir,
So let Untik speak first and then I'll ask.
Okay. Antik, yes.
sir, this what did you say about the full form of ZAP? Zinc
Finger antiviral protein.
proteins. So, sir, these antiviral proteins were synthesized in that 2002 era. That means for which
No.
the evolution
Antiviral
was
protein is a
okay.
okay. It's my
Hmm.
mistake. I did not explain it well. Antiviral proteins, they are present. Finger antiviral protein is
Hmm.
present in your body as an immune mechanism to to fight against viruses. Hence, all the viruses which infect humans, they have reduced CPG content. So yeah,
Okay, sir.
almost
Okay,
all
sir.
viruses.
Understood.
Not, I would not say all the viruses, but anyway. Yeah, Mayuk.
So, in your presentation, you had showed several graphs and several graphical patterns regarding CPG loss and all. There, I saw one parameter known as P. It was showing that P is much less than 10 to the power minus 4, 10 to the power minus 5,
Yeah,
like
yeah,
that. So, could you
yeah,
please
yeah.
explain that parameter P? What does P stand for?
P is the probability. It's actually a statistical test which is done. So when you're comparing numbers, we do t-test. But when you're comparing proportions, we use chi-square test. So there are various tests to verify whether the difference is statistically significant. Why is that important? Because when in biological systems you see differences, even while you are replicating your experiment, you will see certain differences. But whether this difference is statistically it makes any sense or not, that for that we need to do some certain tests. So here I use Mann-Whitney U test for the t-test. And for the chi-square analysis, I use the chi-square test. I mean, that's as simple as that. There are other tests also. We find out certain distributions of while we are dealing with data. We've tried to find out whether it's a Poisson distribution or something like that. So there we change the test to, our liking. So that will be something that you will learn in statistics, biostatistics in specific.
Okay, thank you, sir. Thank you so much.
Okay, I think we can ask Agrodeep to present if there is no further questions.
Am I audible?
Yes, yes, you are.
I think so. Yeah. Mm, just give me a moment. Yeah. Shit. I will just rejoin because there's some issue with the recording. I'll just give me one moment.
Yeah, sure.
Or maybe I can do this later. I don't know. Security and privacy settings. I don't understand.
Are you using a MacBook?
Yes, that's why I don't understand half of these things. This is just stupid. I can't find my own. I don't know where to find this. Windows, Microsoft. See, it's just stupid. I can't share my screen.
It's actually
Okay.
shared.
Is it visible?
Yes, yes.
Yes, it is. But
Yeah.
yeah, it's a weird.
Yeah. So yeah. Hi. This is not going to be a paper presentation. This is just a normal presentation about what we do. And hi, I am Agradeep Mukherjee, and I'm one of your seniors. I am a batchmate of Sanket, and we passed out in 2020. So I did my BTech in biotechnology in Heritage from 2016 to 2020. And then later for my MTech, I joined IIT Guwahati, where I finished it from 2020 to 2022. And right now, for the past year, I am employed as a PhD at the Biochemistry Center under Dr. Joachim Moser von Filzek at University of Heidelberg. And I will just give you a very simplified view of what I work on. So this that you see on screen is a yeast cell, which is a eukaryotic cell. And one of the primary features or the differentiating features of a eukaryotic cell is that it has membranes inside. So yeah, and if you have membranes inside, you need to deform or reform and do all sorts of remodeling of those membranes. And there are like an army of different proteins. And I work on such an army of protein, which is called endosomal sorting complexes required for transport or escort-3s. So there's a cascade of escort zeros, ones, and twos and threes. So I work on the last very protein complex. So what are escort-3s? So escort-3s is basically wait, let me enable my pointer. There's a pointer. Yeah. So whenever there's a membrane remodeling event, there's this classical omega shape formation. And what we now know from various studies is that there are proteins which sit on the neck of that invagination, and then it just constricts and severs the formed vesicle. And escort 3s are the last protein members that are involved in many of these membrane remodeling events. There are other proteins such as dynamines and everything. But yeah, the most important and the exciting thing about escorts is that usually what happens is if you want to constrict something, let's say a mitochondria, which uses dynamin related proteins. So if this is, let's say my wrist is a mitochondria, the proteins will just sit on the outer surface and it will just constrict it. But escorts can do it from the inside. So that was the exciting part. And escorts are highly conserved, so they can be found in archaea and bacteria and even you and me and all the different mammals. So that's the exciting part, and the only energy utilizing thing of this entire complex is this one ATPase. So it's like an entire car and one engine's driving it. Compared to the other dynamines that I told you, they all have their own machinery, like they all are GTPSs or something like that. So yeah, that's the important thing. Now, it's known that this occurs, but the last step, how it actually constricts and forms the let's say severs the bud, it's not known. So I am working on that, and my approach of work is to generate these omega shaped invaginations and later recruit these escort 3s to study how they work. So this is my project overview. So how to form those omega shapes. So it is generally formed using cargo. So I use a synthetic nanocages as cargo. So these are essentially protein nanocages that just form a 60 is it dodecahedron or icosahedron? I think it's dodecahedron. So they form these dodecahedral structures and they act as synthetic cargo, and when they attach to the membrane, they are theorized to be like forming these invaginations, where later these escorts can be recruited to those regions, and we will study it using cryotomography. So yeah, so this is the like a pictorial depiction of the nanocages, and this is what I am expecting. I'm working on optimization of these nanocages now, and later we will just recruit these escort threes. And yeah, so this is the end of my slide, and I just wanted to show you some pictures, the some real pictures of my nano cages. So I don't know what your screen quality is, but these images are quite grainy because it was not a very good quality staining. But yeah, you can see it looks something like this on the negative stain, and here it looks something like this on the cryo electron microscopy, and we are expecting something like this. So I'm working to optimize this process. Yeah, so this is my current project, and this is my supervisor, and this is lab assistant, this is a postdoc, and this is all our department. We um, go on tours and hikes as like normal classical Germans. They love to hike. So yeah, thank you. And if you have any questions, I am open to taking questions, and you can also contact me on LinkedIn if you have any further questions. I would be happy to answer. Thank you.
Antik, I think Antik has a question. Please go ahead.
Yes, sir.
Ye-
Sir, I have one question. You mentioned about negative stain. So, what is negative stain? Can you explain?
Negative stain is basically a type of electron microscopy where we use heavy metals. Like for my case, I use uranyl acetate, which has uranium 238 atoms. And what it does is, let's say you have a surface and you have some of your proteins or something. The uranium atoms will sit on the surface of it after staining. So let's say you have a crevice and the uranium atoms will be concentrated over there. And if you have, let's say, a dome shaped, the uranium atoms will not be sitting on the dome shape. So it's more like a like spreading marbles over a floor and sitting how the marbles are arranged. So and then when we hit them with electrons, this is something that you get this particular. So see, these are aggregates of my nano cages. So it's like highly crowded, and a lot of uranium atoms just go inside, and that's why it appears as black. But if you see the nano cages, they are like small footballs. So let's say there's a football on the ground, and you try to balance a marble on it. It's very difficult. So that's why these are white. So there's very little to no uranium on the top of these nano cages, but it's all over the like around the nano cages. So it helps us to see. But I'm sure there will be other atoms that are used too. But uranyl acetate is one of the easiest way to negative stain. I hope you could see
And
the
sir, one
screen.
more
I'm
thing.
not sure.
Yeah,
Yeah.
and yes, sir, one more thing that you mentioned about these synthetic nano cages to be
Mm,
dodecahedral structures. So,
yeah.
what problems are you basically facing when you are adding means the synthetic nano cages which you are synthesizing the uranium
Mm,
atoms, like they sit on the surface, which blocks
mhm.
the entry points, right? After the synthetic nano
No,
cage.
no, no.
Okay. So that part I cannot understand. Can you please
It's a different thing. See, negative staining is just just a way to visualize the nano cages and their
And what is
interaction with
inside
the
the synthetic
electrons.
nanocage? Means
It's
what is
empty.
inside
It's
this?
empty.
It's
It's
empty.
empty.
Okay.
It's like a football, except that there's no air inside, and it's like a how to tell. It looks exactly like this with nothing inside, but you can pack stuff inside. So there are people who are researching and packing drugs inside, and then uh, with certain like mechanisms that nano cage will travel to your desired place, and then it will just rupture, revealing the drug. So the drug is protected from the body processes. And these are very robust nano cages, I must say. And they're really tolerant. So yeah.
Okay, so the uranium which you are speaking of, the uranium is getting accumulated on this. Suppose means each surface of a dodecahedron is basically a pentagon, right? Huh? A pentagon.
Pentagon, yeah.
So the means if I consider one pentagon, the space which
Mm,
is hollow in the pentagon, that means the
mm.
middlemost part which is surrounded by the edges, that part is
Yeah.
getting like accumulated with uranium, and then
Yeah,
after
I
hitting with
think some of
electrons.
it enters. If I can zoom it, I don't know how
So
well you can zoom. Can you see it? If you see
it's
on
it's
this
visible.
particular
Let me
thing,
zoom here.
if you see this particular thing, you
Yes,
see
sir.
these are the sides and the vertices of the nano cage that are white.
Okay.
But the faces that the uranium
Yes.
can still go inside, and that's why you see such a nice uh, like it's not the best staining that I have done, but this one here was really nice, so I wanted to just show it to you. And here there's no staining, so this is just like normal electrons. This is cryo-electron microscopy, so they don't use staining here. It's just that you freeze the samples, hit them with electrons, and you see how the electrons are like, yeah.
Okay, sir. Okay, sir. Understood.
Any other questions?
That's a really, really nice presentation, Agrideep. Just for the curiosity, I just wanted to ask one thing.
Yeah.
How are these nano cages better than other nanoparticles, like let's say nanodots, which have porosity or something like
Mm,
that?
I am not sure. I don't have a really thorough comparative study how they are better, but
Maybe
if
something
you,
I'm asking about is this structure. How is it? Very important. Uh,
I mean, why there was not a lot of options, but why I chose these particular nano cages is that if you see the size of these nano cages, I think many of the viruses resemble this particular size. So I think
right,
for
right.
a one nano cage, if I zoom in here, it's around 25 to 30 nanometers. And if you enclose it in a vesicle, like a lipid vesicle, then it resembles
Mhm.
something like a viral egress. So we are later planning on studying the viral egress process, and we will use the escort trees because as we know now that Ebola and HIV-1 viruses use this escort tree. They just hijack this escort tree proteins, and they use that to just escape the cell. So if we can find out something about this process in like more detail, then yeah, maybe later we can shed light on these processes because most of these viruses are very deadly. So HIV-1, Ebola, and
Right, right, right.
I'm sure
Makes
this will.
sense. Yeah. Thank you so much, Agrideep. Thank you so much. I would just like to
Yeah.
inform you all, Agrideep has been one of the only panelists, I think, here who has been very closely related with aging. He has studied about mitochondrial dynamics in specific in his master's, and he did that using yeast cells. So I think he is the best one to talk to if you have anything to ask about aging.
Mm,
But anyway, we will move forward to our next
yeah.
panelist. Anonna, I think you can go ahead if you're ready.
Sure. Okay, why can't I share the presentation?
Are you also using a MacBook?
No, but I'm not getting the PowerPoint
Share
as a
option.
tab
Oh,
at
acha,
all.
acha. I think it doesn't come as a tab. You have to click the tab option and then you will get your windows. Uh, you have to click the window in the share option and then you can get.
Yeah, I'm doing that, but yeah, wait. Let me see. Does this work?
Yeah, yeah, this is working.
Yes, it's
Okay.
working. I can see.
Okay. I won't be able to see what is happening here, but yeah. Okay. Okay. Hi, everybody. I'm Ananya Bhattacharjee, and I'm currently pursuing Masters in Biomedical Engineering from ETH Aachen University in Germany. I am a master's student still, so I don't have as much research to show you a paper or talk about a paper like Shanket and Apradeep, but I'm going to talk a little bit more about how to pursue masters abroad if you are interested, or even in India, and how to go about it, how to build your profile, and how I'm here and from where I
Ananya,
came.
just stop. Just apologize for interrupting, but I think your presentation is yet still not visible. Have you started?
Yep. Wait, I'll try again.
If you're having a problem, you can send it to any of us and we can maybe help.
Yep. Is it okay now?
Let me. Yeah, yeah. Now
Yeah,
it's
yes,
perfect.
yeah.
Okay. Yeah, so I'm doing a master's and I'll talk more about how to get here. Yeah, so this is a short timeline of when I graduated high school in 2017, and then I was for 4 years in Heritage Institute, like all of you. And then,
Ananya, again, sorry, you are in the first slide.
okay, why is this happening? Am I in the second now?
We can only see the presentation. Now it's changing. Yeah,
Now
now
it's yeah.
Now?
There you were in the second slide. Yeah.
Okay, I'm in the second slide, right? Yeah,
Mm.
okay. Sorry, I don't know why this is happening. Yep. So I'm in Aachen now, and I hope to graduate in 2025 if everything goes well. This is like a short overview of what I have done so far. These are some of the projects that I did back in India, and I was doing them during COVID, so I had a lot of time to do projects. I don't expect you guys to have this many projects by the end of bachelor's, but I would say try as much as you can to proactively do internships and projects,
Mhm.
because they also are can be used
You
as
are
learning.
Ananya, you are still in the second slide.
Okay, I'm gonna send it to someone and you guys present to the
I
Yeah,
think
government.
maybe
I have
we
it.
can.
I have it with me. I think I can
Yeah,
share.
can you present it?
Just
Yeah.
give me a moment.
I don't know what is wrong with this.
I think they are not allowing European countries to, connect with India.
Just
There is
shut
not
up.
no already is also here.
Just shut up.
I'll
Sorry
just
guys.
download your presentation.
Yeah.
It never
Okay.
rains in Delhi, but today I don't know what is wrong. It's raining so heavily, my network is also like failing me.
It always
You can
rains.
send it to me if you are having issues.
Yeah, I'm gonna send it to you as well.
Ananya, you had sent it to me. Oh, it's here. Yeah. Let me have
So-
it.
And it always rains here. It never stops throughout the year. It's insane.
I'll download it. Just give me a second.
Okay, just you are also downloading it. Okay.
Yeah, you try first, and if
There
if
are a
it
few
does,
works. It's fine.
try first. Yeah.
Did open although. I mean, let me see if I can navigate. Uh,
Ah, yeah, okay. Thank you. You can go to the third slide. Yeah. So I'll talk a little bit about the projects because it's interesting to know how I got them so that you can also follow similar patterns. For the first one, I was looking for internships and I wasn't getting any on field because of COVID. So I started mailing all the professors I could from all IITs and NITs, and I started giving them my CV and a short cover letter, a general cover letter, that I want to work for you or work in this field, etc., etc. But make sure that you don't mail too many professors from the same university because then it gets crowded. But yeah, and one of them from IIT Roorkee replied, and I was a part of his project that was on data analysis and role of beta lactamase in antimicrobial resistance for 1 year, and a lot of my colleagues after me joined as well for that. Then I also would recommend all of you to apply for the IAS fellowship. That is the Indian Academy of Sciences in Bangalore. They provide a fellowship every year, and the form comes out around October, so it's almost here. And you have to give them research proposal in the application where you have to propose or suggest a novel research possibility in your own words, and you are selected based on mostly that. So I got selected for that, and then I worked under, and they allot you a research institute and a professor. So I worked under Dr. Shilpa Sawant from Bhaba. And yeah, we wrote a paper because it was online. I couldn't go, but usually if you get the fellowship, you go to that institute and you stay there for 2 months and work on a project. So it's very nice. Then, because of COVID, a lot of my colleagues weren't getting many internships. So Heritage decided to give us internships in the college itself. So that's where we did bioremediation and enzyme kinetics. And I also followed up on the same research for my bachelor thesis. And then I randomly just went to the HR departments of hospitals because I wanted some on-field experience, and it like mailing or calling wasn't working. So I showed up at the HR department of Apollo, and they were open to taking me as a pathology intern. And there I worked for, I think, 3 months. Um, EduFabrica is not an amazing opportunity. I mean, it comes every year, and you can do some kind of review articles and journal articles under the program if you want, but I won't consider it as a necessity. For people who are thinking of applying abroad for masters, you should know about what GRE and IELTS are by the time you are in your second semester, end of second semester, and then probably try and prepare for them. And GATE, everybody knows. Yeah, I think we can go to the next slide. So what I am doing currently, the facets where I'm working right now include cell culture, image analysis, immunohistochemistry staining, hydrogel formation, drug delivery. So I still have my courses going on, so I'm not doing research as in my course yet, but I am doing a mandatory internship right now, and also I am working here part time as a work student. That is a European culture that you get used to when if you come here. We almost all the students here try to have a part-time job because we need the money to sustain ourselves in this country, and it's also a good learning opportunity and experience if you get a part-time job in your own field, which is not very hard to get. And yeah, so in yeah, so DVE Leibniz Institute of Interactive Material is where I'm doing my part-time job under Professor Lapote, and we are working on a project called Mend the Gap. It's basically a spinal cord injury repair project where we are trying to make an injectable hydrogel that you can apply during, because once if you have a cut injury in your spinal cord, it often happens that that injury doesn't like get back together, like because the nerves are severed and then you have paralysis somewhere or other in your body. And so to accelerate growth and repair in neuronal cells, what we're trying to do is inject hydrogels that are treated with magnetic particles, and then these hydrogels go inside that site of injection, and they align the neuronal cells from both ends according to the direction that we want. Like we use a magnet or a magnetic component to align them such that the neurons in those endings grow in a particular alignment or direction and try to connect the severed connection. The obstacle, current obstacle in this is it's working, but it's working in chick embryos. But the problem is the time management, the time that it's taking to inject the hydrogel and accelerate repair is too much. And when you're ongoing a surgery in a human body, we need as less time amount as possible. Yes, Srin John.
How are you ensuring that the hydrogel will stick together till like for the entire time?
What do you mean as stick together? Do you know what hydrogels are?
Not exactly. That's why I wanted to know about it.
They're basically 3D gel kind of particles, and these particles are treated with magnetic properties. So they are magnetic in nature when we are injecting them. So when we are putting them or like keeping them in close connection with a magnetic component, then the way we are aligning the magnetic component, that way the hydrogels align. They're 3D structures, so they will align accordingly. I don't have a picture to show you, but yeah. So then these hydrogels
Should I
align.
change the slide?
No. These are
Okay.
hydrogens align, and they're also treated with neuronal growth factors. So what happens is these hydrogels release those growth factors in this alignment and then influence the neuronal cells to grow in that direction. So there's no there's no connection to break off. They're influencing. They're not directly connecting with the neuronal cells or making them grow. They are releasing growth factors and influencing the growth. Anyway, moving on. And for my internship, I'm working in the the previous slide.
I thought moving on is going to the next slide. I'm really sorry.
No
Okay.
problem. The internship that I'm doing is in the Institute of Experimental Molecular Imaging under Professor Lammers. And here we are working with cancer activated fibroblasts. And we are seeing if certain cytokines like TGF beta and IL-1 beta can influence or influence in any way the production and acceleration of fibrosis, like formation of fibrosis and collagen deposition in cancer cells, in certain cancer cells. This is one of the fluorescence imaging that shows that when we mix fibroblasts and cancer cells in a spheroid, the fibroblasts tend to deposit in the middle of the cell and surrounded by the cancer cells. So this is marked with vimentin, which is a very good marker for fibroblast cells. And DAPI is the normal staining that we use to stain all cells. So DAPI is in blue and vimentin is in red. And we'll do then the quantification and analysis and see if these cytokines have any effect, and if any, then how they are affecting, if it's good or bad, etc., etc. So these are all ongoing research topics that I'm working on. Moving on. Next slide, please. That picture was how the hydrogels look when you're making them, but then you harvest them and then they become these very small particles of some micrometers, 20 micrometers. Moving on. So
How are you making them?
we are making them using a thing called PDMS. It's a compound, and you treat them with certain chemicals and also the spions for magnetic properties, and then you treat them with UV, and then you have different techniques to harvest them, but that will become another talk entirely. So for the tips and
PDNS.
PDMS, yes. There are also different kinds of hydrosols, so it isn't just PDMS. There's sawtooth-based gels and other gels as well. I think Shashwato knows better than me about this, even though I am working with it. But yeah, for tips and tricks, I just wanted to tell you that like if you want to apply abroad, then it's better that you start deciding on your career path, like what kind of field you want to go to. It doesn't have to be specific. I didn't have a specific field, so I chose biomedical engineering. But if you have a specific field in mind, you can also search in that area, for example, immunology or viral something or oncology or whatever. Then plan well ahead of deadlines. Don't keep things for the last moment, because then it usually never works out. And build a strong directional profile. Ask people, ask seniors, ask people who have already gone through this and are studying outside or doing their PhD and stuff. And try to have as many projects, courses, work experiences. Online courses are also like pretty good if you know which courses to do, and they also help you have a basic idea of things that you might not learn in your 4 years of bachelor's or do not have enough of in your bachelor's. Also, please, this is very important. Learn the difference between statement of purposes, letter of motivations, and cover letters. These are very different three things. And I often get juniors who are asking me, what is the difference? How am I going to write about this? I don't know. This seems similar to me, but there are very particular differences that you should know about. Yeah, network well with alumni and take internships and projects as a learning experience. I think I talked too much, but I hope you get an idea of what to do next. Thank you. If you have any questions, except for very detailed project related questions, because I am limited in what I can share since it's not my project. But in general, I can tell you.
How
You can
much
tell about
temperature
the project.
varies in Germany? That's a good
What?
question.
What?
How is the temperature there in Germany?
It's insane. Like right now, it's I think 21 or something, but at 6 or 7 in the evening, it's going to be 16 degrees. So it varies by 10, 12 degrees every day. It's crazy.
It's similar to Guwahati. We get like a heat shock for 15 degrees. In the morning, it's
Crazy.
15
26,
degrees is a heat
27.
shock for you.
Yeah, right now it is
One
for me.
five,
Right now it is.
15
Right now,
degrees. 15 degrees is a heat shock.
yeah,
It's
right now it is
you
for me.
don't understand when you live in minus degrees for months and then you have to live in
Yeah.
23 degrees. It feels like you are in a desert.
Also, Indians are well equipped at their homes for the heat. They have fans, they have everything. But here, the people just wanted to conserve heat, and now
Exactly.
it's so much. I mean, hot inside. There's no
It's
fan, nothing. So it's
There's no
terrible.
ventilation. There's no concept of
Yeah.
ventilation in this country. Like
Yeah, they just
the
wanted to
classrooms
conserve
are
heat.
just yeah. Yeah, there's no ventilation. Like summer is really, really torture here. Like it's worse than India. And even though the
And
temperature is not that high, because
even
you
though
have
it's
no
25 degrees, but it is worse than India.
yes, because you have no ventilation. There's no exchange of air in here anywhere. There's no fan. There's no forget about air conditioning. There's not even any fan anywhere. It's crazy. And you always have to be prepared for every kind of weather. Like it can rain, snow, and be like very, very hot on the same day.
Be
Not
sunny on
even
the same day. Yes.
yes.
It can snow in the evening. You never know.
Yeah. Like within a span of 2 hours, the temperature goes from 4 degrees to 18 degrees. It's crazy.
Okay, okay. I
Anyway,
think
just
uh,
yeah, let's do
any
this later.
questions? I think we will move on with the next presentation. Orko, what about food?
Yeah.
We will give you a complete booklet about it, Shinjon, food and spices and everything. You can decide on it. Let's move
You
on.
can also approach us anytime, okay? Like I think I speak for all of us in this, that you can also like ask us questions anytime.
How?
Orko, are you with us?
Yes, yes. I'll
Yeah,
just
please.
share my screen. I hope I don't have any problem. Is it
Yeah,
visible?
yeah. It's visible. It's perfectly visible.
Okay. It's is it full screen or I can't? I
It's
think
full
I.
screen. It's full screen.
Okay. So yeah. Hi. So I'm Orko Roy Chaudhary, and like others, I'm also an heritage alumni. And so I did my BTech in biotechnology from 2018 to 2022, and I'm currently in my second year of masters, and I'm doing my MTech in bioengineering. So from the Department of Bioengineering, so the name was like changed almost 3 days ago. So we don't have a logo yet. Earlier it was called Biosystem Science and Engineering, and it's an interdisciplinary department where people from all computer science, electronics, mechanical, from everywhere they come together and work on interdisciplinary projects. So I'll tell you about my MTech project a little bit. I've just got started from July. So I am currently in the mechanobiologics lab under Dr. Ajay Tijore. And so we work in the interdisciplinary field of mechanical forces and biology. And to tell you a bit about the background, like our lab focuses on cancer mechanobiology and stem cell mechanobiology. And currently, I have got two projects undergoing, one in cancer and one in stem cells. So my primary project is under in cancer mechanobiology. Moving forward, a little bit of background about my project. So we know that the cancer stroma or the tumor microenvironment comprises of ECM, stromal cells like fibroblasts and immune cells, and it is very important for tumor initiation, progression, and also tumor invasion. So the cancer, so the fibroblasts of in the tumor stroma are called cancer-associated fibroblasts, and they can modulate cancer progression as they secrete most of the ECM components. They are can remodel the ECM and help in the epithelial to mesenchymal transition, which helps in the invasion of cancer. And they secrete various biochemical cues like growth factors and cytokines, which prevent the immune cells from targeting the cancer cells. And like my PI did the work my PI did in his PhD showed that cancer cells are vulnerable to external mechanical forces like ultrasound. So we work with ultrasound in our lab, and confinement and stretching of cancer cells. So they are not very well known to adapt to mechanical forces, and they undergo apoptosis. So I am like pursuing the avenue of like what is the effect of mechanical forces on cancer associated fibroblasts, because they are also transformed cells, like the fibroblasts are also transformed, and the wound healing properties of the fibroblasts have been hijacked by the cancer cells. And so I'm currently investigating the effect of mechanical forces on the cancer associated fibroblasts. So this is the title of my project, and I'll give you a brief overview. This I presented the same thing on my research proposal, so I'll just walk through it. So we have collaboration with a local hospital here, Ramaya Hospital. You might be knowing, it's a very famous government hospital here. So we have a collaboration. We get patient samples. So I work with primary cells basically, and we get oral tumor sample, and then we isolate cancer associated fibroblasts from the oral sample and treat it with ultrasound to see the effect. And I've already done a couple of experiments, and the results are quite positive. Like we are seeing apoptosis. This figure was from 1 month ago, so we didn't know whether the calves would survive, grow. So next we'll do the mechanistic study, like what is the focal adhesion. So the focal to be brief, like we in BSSC see cells as a machine, like the forces they experience, and what happens when the substrate is a bit rigid and the substrate is a bit soft. So focal adhesions are basically the components at the periphery of the cell when they interact with the ECM. Okay, so we will currently study the focal adhesion of the cancer associated fibroblasts. And my future goal is to create a calf and oral cancer cell 3D spheroid, and also culture the oral cancer cells in the conditioned media of the calf. So conditioned media, I can tell you, is basically after the calves are treated, so they stop secreting cytokines and chemokines because they are altered now. And so in that media, how the cancer cells are behaving, that is what I'm going to check in the future. And also this 3D culture or 3D spheroid, I'm going to treat again with ultrasound, and then check their behavior, whether it helps in the apoptosis of cancer cells. And then the future of this project, which is beyond the scope of my 1 year MTech project, is in vivo and clinical testing of this mechanical force as a therapy. So this is about my main primary project. I am also currently doing a second project, which is the activation, like induction of human mesenchymal stem cells towards osteogenesis using ultrasound mediated mechanical forces. So stem cells are very well known to take the mechanical forces. So during cell signaling, if you have taken that, you had a compulsory cell signaling course. So when mechanical forces are introduced to the stem cells, there is MAPK and ERK pathways that are activated within the stem cells, and they go, and the downstream effect of those signaling proteins are the upregulation of osteogenetic factors, transcription factors or osteogenetic markers like osteopontin, RUNX2, BMP. So I'm currently studying that area, whether continued whether continued mechanical forces at a at a particular interval of time, they can if they can undergo osteogenesis. So this is the concept figure. So we have isolated human mesenchymal stem cells from bone grafts, and then we are checking the osteogenetic markers and after they have been treated with ultrasound. So here we have to do RT-PCRs, and then we also wanted to check what is the difference in the expression level of osteogenetic markers on different substrates. So of different stiffness and different substrates. So we make hydrogels of different stiffness, and then we culture cells on them and then treat them. Like this is not treating, and we induce them to mechanical forces to check the osteogenetic markers. And next, in the future of this project, we'll also try to make a 3D bone graft using this, like culturing stem cells in 3D, and then if we are successful in 2D, then treating them in 3D, and if we can make a successful bone graft through by culturing those stem cells. So yeah, these are the two projects that I had to share, and yeah, that's all. If you have any questions, and in the first slide, I have given my mail and LinkedIn. So if you need to contact me, you can. I'll send this presentation to Sanket. Yeah.
Lots of questions.
Yeah.
Antik, you may
Okay.
go ahead.
Sir, can you explain what is osteogenesis? What is the
So
meaning of this term?
osteogenesis is basically formation of bone from stem cells. So stem cells, can differentiate into multiple number of cells, right? So osteogenesis is the formation of bone cells from the human mesenchymal stem cells, and this can be due to bone injury, or there are osteoblasts and osteoclasts. So osteoclasts are there for bone resorption. So when a bone cell undergoes disintegration, then stem cells form bone cells. So that process is called osteogenesis, like differentiation of stem cells towards forming bones.
Okay, sir. Okay, sir.
Shinjan, I think you have something.
What's the role of ultrasound here for the same?
So like ultrasound's role is mainly to provide mechanical force. Ultrasound is basically vibrates at a higher frequency than the audible range, and so basically they are not they uh, provide a shock to the cells. Okay, so cells, generally cancer cells don't take mechanical forces that well because they have altered calcium homeostasis. So because of that, they undergo calpain mediated microtubule depolymerization. So that's why cancer cells undergo apoptosis. That's the main logic behind
So can
ultrasound.
you
So we
elaborate this a bit? This thing,
elaborate on what?
why cancer cells does not go on apoptosis? What they develop
So
that part?
so basically, piezo one is one and is the mechanosensitive channel. They are generally present in the cytoplasm, and basically when there is an external mechanical shock, it can be, suppose you're hitting someone with a force, that's a mechanical shock to the cells. Okay, so any extra mechanical force which is not generally present within the body is known as a like external mechanical force. And excuse me just a minute. Yeah. Okay. Basically, what cancer cells do, they have an altered expression of piezo one. Okay, and an altered calcium homeostasis. So in normal cells, there is calcium homeostasis, and the calcium level within the cell is well maintained. So calpain is a cysteine protease. Yeah, it's a cysteine protease, and basically these are calcium dependent. So if there is more amount of calcium within the cancer cells because of this altered calcium homeostasis, so the calpain is activated and they basically disrupt the microtubule, which helps the like cancer apoptosis.
Okay, I think that's before we get to
One
human.
more just what are the alternatives for this ultrasound?
Anything. We are currently investigating two more forces, which are confinement. Like one of our PhD students is making PDMS channels, which like they are. So, what is the effect of confinement on cancer cells, and also stretching of cancer cells? So, stretching, ultrasound, and confinement. These are all the mechanical forces that we are currently investigating and their effect.
Sir,
Yes, Mayuk, please.
sir, could you please explain the part C, that is molecular mechanisms at different stages of osteogenetic development here?
Yeah, so basically I'll say first, when the stem cells, there are some early osteogenic markers, there are some middle osteogenic markers, and there are some late osteogenic markers. The late osteogenic markers are obviously the bone cells, which are the osteoblasts. The middle osteogenetic markers are the osteopontin, and the early osteogenetic markers are WNT, BMP, and RUNX2. So basically, we'll check because so in this project what I'm doing is treating the like, well, I'm very used to the term treating since we under like since we work on therapy. So basically we are making the stem cells under mechanical forces at different intervals of time for 14 days, every day for 14 days, and then we are checking the level of osteogenetic markers at different stages of osteogenesis. So osteogenesis is a long process and it takes months. So basically, we want to check at the different stages of osteogenetic development, and molecular mechanisms means just the assays that we'll perform, will the proliferation tests and basically RT-PCRs and Western blots to check all the different markers. This
Okay.
is basically a concept figure that I use.
So,
That's
here, what do we mean by HMSCs, which are undergoing the two kinds of pathways, the various kinds of pathways, as we can see here in the slide?
HMSCs are human mesenchymal stem cells, and I'm telling you again, like, don't go into this figure. So basically, they are undergoing two pathways because, like, this is not two pathways. This is the two avenues that we are checking in our project. So one is the cell contractility pathway. Like when we induce mechanical forces, the cells will contract, right? Right, like they won't stretch or spread along. So under that contraction, so there is YAPTAZ pathway in the down regulation, like in the downstream, which helps in the osteogenic markers to form, which helps in the formation of bone cells. And the transcriptomics means all the analysis of the transcription factors. That's it. This is all. This is about the project. And HMSCs means human mesenchymal stem cells. We derived these from bones.
Thank you, Orko, so much. Just a quick, just I would say something like I was reading an article, not a research article, but rather a normal news article that cats they purr also. They when they purr, they make some ultrasonic waves and they have bone healing properties, something like that. There might have been
Yeah, I
some
have
studies.
read about it too, and yeah, I know.
So yeah, I mean, this looks like it is aligned with that. But anyway,
So
as you are from
we can't use cats for
right.
project,
Right.
unfortunately.
But definitely, since you are from IISc, I think you have the most voluminous work out of everybody else who's present here. So anyway, so thank you so much, Orko, for giving
Thank you.
some
And
time.
again, like, sorry for the like, there are no details in the slides because I just made it 10 minutes before this meeting. So
Yeah,
yeah.
yeah. Okay.
Okay. Thank you.
I think Shashwata, you are next. We are because of the interest of people, we are running late today. think we have to extend this to some other day as well.
And we all did, Orko, we all did.
Oh, okay.
Is it visible?
yes, 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 yes.
Okay. So, good evening, everyone. I am Shashwata, and I have also done my BTech in biotechnology from Heritage. And I am a batchmate of Ananya and Orko. And now, currently, I am pursuing my master's in biomedical engineering from NIT Rourkela. So, I have shared my email ID and LinkedIn. So previously, I have worked on like various projects which involve like bioremediation, microbiology, development of nanoparticles, also some work with cancer, like assessing the role of certain enzymes, and the other vaccine technology thing that we all most of us did during the COVID. So the key skills that I learned during all of these years, recently I have learned cell culture and a lot of cell-based assays. I think it is key to pursue good research. You need to have a very good idea about cell culture. Material characterization is something that is important to my work, particularly since I'm working with biomaterials. Uh, and as a part of my coursework, I have also learned Python to analyze biomedical signals since it is a core part of biomedical engineering. My previous experience with microbiology is a skill as well, probably. And I enjoy Adobe, Photoshop, and Illustrator. I learned it during my BTech years. So I'll keep it very short. So my current areas of focus, currently I'm pursuing two projects. So the first one is the 3D bioprinting for skin tissue engineering. So let me explain what hydrogel is. So hydrogels are essentially, we take polymers and we cross-link the polymers together in order to form a gel-like substance. So essentially these polymeric networks can entrap a lot of water within them. So there is a transition from like a liquid to a solid form, a sol-gel transition essentially. So that is essentially a hydrogel. So it is basically a cross-linked network of polymers. So what I do is I encapsulate the cells in the polymeric matrix and I obtain a bioink. So in case of 3D printing, we use PLA and other filaments in order to print certain structures. I hope all of you have a good idea about 3D printing since it is a very new and very exciting technology. So what I make is I make polymeric scaffolds for skin tissue regeneration. And my current goal is like a lot of work has already been done in skin tissue engineering and making bioprinted scaffolds as well. However, I am currently addressing the drawbacks such as tissue adhesion, and also I'm trying to uh, fabricate a bioink as such that I don't have to incorporate any other like other growth factors or antibiotics or any other drugs in order to improve their inherent wound healing capacity. So I'm exploring polymers and as such, mostly biopolymers, which have inherent wound healing properties, and which my next work, but in the next semester, I will start work with stem cells as well. So I want to see whether my scaffolds have the inherent property uh, to let the MSCs go from from the stem cells to fibroblast lineage only. So that is what I'm aiming for. The bioprinting method I use is an extrusion-based bioprinting where we use a piston and the piston pushes down the bio ink through a nozzle and we can print particular scaffolds that we can design on our based on our interest. So you can use any CAD modeling softwares to design particular scaffolds. On the other hand, when you are dealing with disease models, you can like suppose we have a very irregular wound. So we can take a CT image of that and we can put it as a CAD model. We can feed it to the bioprinter and then we can print that exact structure so that it fits properly in the wound itself. So this is a basic overview of the 3D bioprinting part. The other project that I'm working with is a fast clotting hemostatic powder. So hemostasis is essentially what it means is clotting of blood. So a lot of commercially available powders
are present. However, what I am aiming for is a waste to biomaterial thing. I cannot reveal what I am converting since it is a very novel method. So I am taking a waste and I'm converting from that waste. I'm taking some biomaterial and making a powder of it, and I am trying to check whether it has a proper hemostatic property or not, or whether it can rapidly clot blood. Since the thing I'm focusing on is gunshot wounds, so the powder should clot within at least like within 1 minute. So that is the time frame that I'm focusing on. So these are the two projects that I'm currently working on, and this is a brief idea about how bioprinting works. So essentially, you can see that the nozzle is printing a structure in this video. So let me. So this is a nozzle. So this is a nozzle. Yeah.
No, you can go ahead. Sorry for that.
Sorry. Yeah. So this is a nozzle that we are using, and this is my bio ink. And I have previously designed a structure in a CAD model, and I'm printing that structure itself. So what I use is not essentially a bio printer. It is a modified 3D printer in which it has been like we have attached modules for bio printing itself. So this is the polymeric scaffold that I printed, and this is the FSM image of that, where you can see there are a lot of pores, there are a lot of fibers in this image, and that is what we are aiming for. We are aiming for a fibrous scaffold which would mimic the our natural extracellular matrix. So the components that I am using are also present in our extracellular matrix, so it can rapidly induce tissue rejuvenation. And this is a live date assay. So the green things that you are seeing, green dots, these are essentially the live cells inside the scaffolds. So when we print, we encapsulate cells within the polymeric matrix itself. And then we print. So you can see a proper filament shape in this image. So the cells are inside the filament, and the thing is that they're living. So that is what we aim for, that the cells would continue to live. And this is the day 14 confocal image of the scaffold itself. So the next thing is this is the powder that I'm using. So it is also a FSM image. So you can see there are fibers, there are powders. So it can trap blood very well. It has a good absorbance. It can absorb blood quite well. I think it is around, it can absorb 48 times more blood than its own weight. So as you can see, there is a clot of blood formation. I am currently optimizing the clotting time. So this is what I'm working on right now. And hemolysis is like, just to give you a basic idea, we cannot use biomaterials or polymers or chemicals inside the body, which would like literally break down your blood cells, right? So hemolysis is a standard assay that we use to check hemocompatibility, whether it is compatible with the blood, or if it is not, then we have to change the composition and or like, reduce its rate. So this is a basic idea about my work, and this is life at NIT Rourkela. These are all your seniors from Heritage, and this is my lab group. This is NIT Rourkela, and it's fine. So that's it. Any questions?
So can you go to
Before
slide 5 once?
okay, just Shinjan, just give me a moment. Before we get forward with any questions, how many of you are ready for presentations today? Please raise your hands because we are already beyond time. Anushka Mayuk, are you from the same group? Ingeet, are you from the same group or you are from different groups?
So Anushka and Ingit are from the same group, and in I'm presenting individually.
Okay, Srinita is also there. Can some of you maybe keep it to the next Saturday? Maybe will it be possible? Because I don't think it's already too long. Uh, is it okay for
Yes, sir. It is okay. Next Saturday.
It's okay.
So I can present today.
You can present today. Okay, we will take one presentation. Srinita, is it okay with you if you present next Saturday, or do you want to present it today itself?
No, so next Saturday is just fine.
Next Saturday is fine. Okay. So after this, Mayuk will take up with his presentation and then we will meet next Saturday. Probably your panelists won't be available next Saturday. But anyway, if they are available, you people are so interested that it takes a lot of time. So I think we can have the time which we have left for you to ask them questions. So yes, I think Shinjan was there with some question.
Like slide number 5 once, and the second picture, I just wanted an explanation. Like, yeah, the five
This one.
evaluation of clotting time. Yes, sir.
So what we do is we take a small amount of a sample and we pour blood on it and we check how much time is it taking to clot. So if it is flowing down, then the clot has not been formed. And as you can see, like all of it has clotted in a single part. So the blood is clotted properly. So it is essentially how you evaluate clotting time, like simply.
Like, how is it clotting? Like, outside the body, like kind of. I
So
mean,
see, the thing is that since my powder has a good absorption property, I add blood accordingly so that there is some excess blood that the powder can clot and I can check that the blood has clotted. So this is essentially the excess blood which has not been absorbed by the powder and it has clotted.
okay. Okay.
Sir, I
Yeah.
mean, Sanket sir. Not
Yes, yes.
sir. Actually, like, because of the time crunch, is it that sir we cannot present our presentation today, like for the
Yes,
time crunch?
actually. Yes, yes. It can't go after 5 like that. I mean, it has been too long, right?
Oh, sir. Okay, sir. Sir, actually,
You
like,
have anything to say?
sir, like, after Mayuk, if there is time, then I can present, but I
I
don't
mean, if
think.
there is time, then I think you people can discuss with the panelists. We can have an informal session like that. I thought it would be better than having a presentation again. I mean, if you want to present, then we may have you as well.
So if time permits, then I'm there. Otherwise, it's absolutely fine, sir. Then next Saturday.
Okay, okay.
Sir.
Thank you, Shashwato. It was really, really nice. You people are working a lot harder than we ever did. But anyway, all the best. Think you'll have a good patent out of this.
I hope so. I'm just trying to finish it by the next few months.
So yeah. Any questions? If none, then we'll Mayuk, you can present.
Okay, sir. Okay, sir. I'm starting. Sir, is it visible?
Yes, yes.
Sir, I'm beginning.
Yeah, sure.
Okay, so good evening to everybody. I am Mayuk Mukherjee from the Department of Biotechnology, third year. I will be presenting a a presentation about the research paper relationship between aging and late progression of Parkinson's disease. It was a clinical pathological study which was conducted in 2010, but the major part of the project began around 2001 to 8 in that period itself. So I'll begin with the presentation. So these are the contents. This is what I've made in the presentation. We have the introduction coming up, following an experiment, and then the discussion, and finally acknowledgements and references. So as we know that Parkinson's disease is a disease which is actually it's actually not a microbial disease. It's actually a disease of the nervous system itself. It's a neurological disorder where the person is symptomed with fatigue and motor nerve problems and etc. So there had been a motive to investigate the relationship between age, advanced clinical stages of Parkinson's disease, and neuropathology. For this project, 129 case records were surveyed under a consensus from donors with pathologically proven Parkinson's disease, and this experiment was conducted at the Queen Square Brain Bank. It's a very important place to be noted for studying the project. It was conducted at the Queen Square Brain Bank for neurological disorders. The cases were separated into 5 groups as per the age at death. Now, there were several symptoms observed in the patients, such as Alzheimer, neurofibrillary, or amyloid beta plaque pathology. This was accompanied by visual hallucinations, which is a very important symptom to note down in the study of Parkinson's. It was also associated with high cortical Lewy body scores. So here it's actually an elaboration of the theory about the experiment, the consensus that was carried out. These are all the different symptoms that were seen in the patients, such as prodromal hyposmia, autonomic symptoms, rapid eye movement, sleep behavior, etc. Parkinson's disease is known to spread by a prior lag mechanism. This involves permissive templating in accordance to the Bragg hypothesis. This is actually a mathematical simulation of Parkinson's disease. That this paragraph actually describes about how Parkinson's disease actually spreads among human beings. So this was a brief summary of the topic. The aim of the clinico-pathological study was to differentiate processes intrinsic to Lewy body pathology from the influences of age, along with reconsideration of theories on the spread of alpha-synuclein and other pathologies. So now we proceed to the experiment itself. For the experiment, around 2001 to 8 patients with initial diagnosis of symptoms as well as pathologically evident diagnosis of Parkinson's disease were identified from records of donors to Queen Square Brain Bank. Eligible cases were selected by scrutinizing clinical and pathological reports held on a database having clinical records. So the ones showing Parkinson's disease, showing evident Parkinson's, were finally selected for the consensus. These patients were categorized into 4 categories. Frequent falling means the ones showing multiple falls during the course of the disease. Visual hallucinations, persistently formed visual hallucinations were observed. The patients diagnosed with Parkinson's disease were observed to see visual hallucinations in a persistent manner. The third category is cognitive disability. In this category, the patients having Parkinson's were physically impaired. They could not perform daily tasks or daily chores in the home using proper limb movements and all. Their limb movements were all improper and basically they were all physically disabled. And the final category is the ones who were admitted in nursing homes and hospitals. These patients were diagnosed in hospitals and were given private treatments under medical superintendents. Finally, the review was found out. It was formulated out of the consensus. Here we can see the first graph where disease course and disabilities are aligned at the time of death for 5 age at death categories. So in the x-axis we can find the age at death, and in the y-axis we can see 2 columns. One is showing the n value, that is the number of patients who were found, and the other side of the x-axis of the y-axis is showing the age during death at Parkinson's. In the following graph, fine lines represent regular falls, dark lines represent residential care. That is the percentage of residential care that was provided to the diagnosed patients. Fine dots here, as you can see in the graph, represent the ones with cognitive disability, and finally dark dots represent the ones with visual hallucinations. Error bars show standard error in the following graph. In the second graph, the sample of 129 cases, as I had stated in the introduction, 129 patients were taken for the consensus, and the sample of 129 cases in the second graph is divided into 5 roughly equal groups according to the age at onset of the disease. The age which is given in the x-axis, or um, or more correctly in the y-axis here, represents the age during onset of the disease. This graph shows the disease course and disability for 5 age at onset groups. Symbols used here are the same as that of the previous graph. Fine dots, dark dots represent the same as there in the previous slide. Now, a keen observation that we need to focus on is that during this consensus, about 9% cases, 12 out of 129 patients, were found to reveal a serious ailment not related to Parkinson's disease. This disease was an unknown disease which was not related to Parkinson's and was completely exclusive. It led to premature death. These patients had a short mean disease course of 10.1 plus minus 5.5 years. That is approximately 5 to 15 years was the mean disease course for these patients. They tended to be younger when they died. That is, they showed immature characteristics when they died, the mean age being 71.2 years, which is approximately 71 years itself. 5 of them died out of neoplasia, 5 of them from cardiovascular disease, sudden cardiac death, etc., and the remaining 2 died from stroke itself. Dopamine receptor agonist drugs had been prescribed to 48% of the patients having visual hallucinations only. Out of these 48%, only 7 patients achieved negligible dopaminergic therapy during the disease course. Provided out of these 7 patients, 6 of them had visual hallucinations. 6 of them showed the symptoms of visual hallucinations. So finally, we come to the discussion point of view.
Mayuk, just a minute.
Yes.
You're over time actually. Please try to wind up.
Yes, my presentation has ended. This is probably the
Okay,
last
okay.
slide.
Sure,
I'll
sure,
wind
sure.
up showing the last few slides, and finally I'll end it. So in the discussion, we can see that our observations can be applicable in reality. Frequent falling, visual hallucinations along with early cognitive decline must be contradictions for deep brain stimulation surgery or for cell-based therapies. Finally, a schematic graphical representation of exponential clinical progression over different disease durations is plotted. This provides the information of how clinical disability varies with respect to course of the disease in years. So with this discussion, we conclude the experiment about getting the information of how the clinical disability due to Parkinson's varies with respect to the course of the disease in years. And finally, this is my acknowledgement to Sir for guiding us all throughout the course
Wait,
and
wait, wait.
for
I have an
providing
issue
the
here as well. It's not Dr. Shanket Mukherjee. Please correct it. It would be missed.
Okay, okay, sir. I exaggerated a bit, sir.
Yeah, yeah. Yeah, please
Okay.
continue.
And finally, this is the references. This is the reference that I had taken. This is a research journal article that was published and issued during 6 June 2010. It says relationships between age and late progression of Parkinson's disease. It was a clinico-pathological study conducted by Peter Kempster, Sean Sullivan, Janice Holton, Tamas Reeves, Andrew Lees. And with this, I want wind up with my presentation. Thank you, and now I'm finally open to questions. Yes, I'm open.
Okay, Mayuk, few things. Too much of writing in your slides. Um, and you went over time. Coherence was becoming an issue. It was difficult for us to understand what you are trying to. Let's go to one of your. Let's go to your result slide. Please go there where you were telling about the lines, fine lines, and there somewhere.
Yes, sir. Okay.
Okay, by this figure, am I supposed to wait? It is okay. Am I supposed to understand that residential care is one of the best way of caring for Parkinson's disease? It looks
Yes,
like
sir.
that, isn't it?
Yes, sir. It looks like that since we can see the alignment of the dark lines in the graph. So,
But
according to the representation,
so when you're presenting things like this, as I told a couple of people earlier also, it's better if you make these observations in your slides. I think that would be from my side. Anything else from the panelists?
Hi. I have the same suggestions as Sanket because there's just way too much text. Sometimes it becomes difficult to follow what you are saying. So instead of giving this much text, you can just organize them into smart arts or like bullet points so that it's easier to follow. And especially, let's say, if you are having like three or four numbers, like 53% of this happened and 75% of this happened. So if you just condense it down to a table with the number and its outcome, then it's much easier. And also, yeah, if there's a figure like your current slide, I would suggest to keep the figure centrally. So the figure should be in the middle, and just a little bit of description of the figure below it, and rest of the things try to just explain it to the audience without just like putting everything on slide, because as a general audience, if you just speak somewhere and you have put in everything in your slides, then you have nothing new to give from your own side. So people will just read the slides and they will just ignore whatever you are saying. So it's better to just give them an overview of everything in the slide. But for the explanation, they have to listen to you to become the storyteller. So you should tell a story in the presentation. It's not like you're presenting data and you have accomplished things. So just keep that in mind. Yeah, thank you.
Yeah, I think Pradeep summed it up pretty well. Same suggestion, Mayuk. This is a lot of this is just text. So maybe this is one of your first times doing a presentation, but usually when you're doing a presentation, it's more what you are saying than what is there in the slides. So as he said, that use the slides as a support system for your like talk rather than just reading out from the slides, because no one will really be, you know, attentive enough to listen to you because they can already see everything in the slides and they can see the entire paper in the slides. And also yeah, use very less text, like use text just to give context about the slide or the images that you are using, and talk more, explain more from your side rather than just this.
oh, oh,
Anything else? Okay, I think this will be the end of today's session. Thank you very much for joining in. You people have really done very well, apart from some negative comments that you have received, even though they are negative, but it's a good opportunity for all of you to improve on your future presentations, because very soon you will be giving presentations for interviews abroad and even in India. So this is very important that you are getting some knowledge from some of your own seniors who are currently at the top of their fields. So having said that, I'm now opening the floor for any informal discussions, any questions that you might have for the panelists. And for the rest of the people, we will be again meeting on the next Saturday on 3 to 5 p.m. same time. I don't know whether your panelists will be able to join, but they are welcome to join if they are available. But I know they are very busy people. So if it is difficult for you people to join on the next Saturday, it's fine. But if it is possible, please do. Yeah. Any questions from anyone? Any of the students?
So can we get your contacts?
I think you have to mail them, or you have to reach through LinkedIn. I can maybe give you the entire list if you want.
Yeah, or maybe you can also send them our presentations. We more or less every one of us has contact info in our slides.
Yeah, that would be best.
Or we can also write it here in the chat.
Yeah, where is
If
the
you want.
chat? I can't find the
I
chat.
I I can't find the chat exactly.
I don't know.
So
I
you
mean, this
need
is
to
something.
log in actually. So you need to log in from the Microsoft Teams, then you will be able to get the chat.
I am logged in through Microsoft Teams,
Yeah.
but I
So
Maybe
now
yeah
the
I'm
guest hole hoy na
maybe
sir.
Shankar can just write
I'm
all of
a,
our emails.
yeah
I'll
I-
send them. I'll send it to them.
okay.
Any questions further, or shall we call it? Let me stop the recording first of all.